Hello, thank you for coming to Hesperos company presentation for Bio International. Hesperos is a startup biotech back in 2014 by Mike Schuller and I. We licensed about 20 patents between Cornell and University of Central Florida. We won the Slush Prize for alternatives to animal testing. We have multiple NIH grants to develop the technology. And we have about 27 employees, and we just moved into a brand new 14,000 square foot state-of-the-art facility. Team, again, I might mention Michael Schuller, considered the founder of the body and chip field, National Academy member, over 75 publications in this space. And I've been working this area also for about 25 years. And that's your real value proposition by working at Hesperos, is you have the expertise of Mike and I for many years. In addition, we have Torsten Hoffman, who is Senior Consultant for Drug Discovery, used to be Head of Medicinal Chemistry at Roche, and Yvette Bilocorta is our Corporate Secretary, and will be soon joining us as Executive Vice President for Business Development. So for those of you who aren't very familiar with HumanOnAChip-based systems, years ago Mike said, look, I can take these equations that are used to try and predict a dosing in a phase 1 clinical trial, adapt them to be able to then try to build a body-on-a-chip or human-based system. This is one of our latest models here, which is a 5-organ system, which is used as part of the HEAL initiative by NIH to look at opioids, to create an opioid overdose model, and then be able to then try to rescue it in the system. These are just all the references that were used to, the basis for this chip. One of the key things that Hesperos has is that we operate in a pumpless system. So, all the tubing and pumps you see off of a lot of the systems, we don't have that. It's enabled us to get rid of bubbles, but also make it a low volume system. So if we have a competent liver, we can actually look at metabolite as well as parent in the same system. The other thing we've been looking at is functional readouts. So when you go into a doctor's office, he doesn't immediately, take your blood for looking at biomarkers. He says, how are you walking? How are you talking? He takes your reflexes. He listens to your heart. These are what we all call functional readouts. And we try to reproduce these functional readouts. We don't really try to focus on cell death. What we want to do is what happens between cell health and cell death, and that functional changes that you actually want to be able to see during a clinical trial. It also allows us to look at non-invasively look at these things, chronic monitoring, and substantially reduces the number of biomarkers we have to actually use. It's kind of a busy slide here, but it kind of goes through how we kind of developed the technology. This was published in Biomaterials in 2015. What we can do, we can create one chip to look at electrical activity for cardiomyocytes. What we do is we pattern cardiomyocytes over microelectrodes. We look at rhythm generation, conduction velocity, and AP length. And in a separate chip, what we do, we actually put down cardiomyocytes on top of a thin piece of silicon. So when the cardiomyocytes contract, they bend it. We can actually look at that position change to be able to then figure out the force. Again, it's our whole focus is try to get function, not reproduce the anatomy. If we can reproduce the anatomy, or if we have to, we can do that, but it's not our main goal in the systems. We're trying to make them as simple and as cost-effective as possible. Again, by patterning the cardiomyocytes, we get the sarcomeres to line up, whether on patterns or on the cantilevers. And again, whether we have a short path length or a long path length, we get the same conduction velocity. This allows us to look at a spontaneous conduction velocity, but also at stimulated. Stimulated makes it a much more reproducible vector. And we can also look at QT interval at every electrode along the way. We've done well over 50 compounds to look at cardiomyocytes. We're going to focus on 3 here to illustrate the general functions of the system. We can look at norepinephrine and show that it speeds up the heart, which it's supposed to do in a dose-response manner. We can look at verapamil, which actually causes a calcium channel blocker, which decreases the force and increases the frequency. And look at sotalol. Sotalol causes a lengthening in the QT interval, which you want to be able to monitor that for cardiac health, but doesn't do anything to force. So again, we can look at mechanistic aspects in terms of saying that pathways involved in electrical activity were probably involved in this compound, okay, but it would not be involved in the pathways for force. We can then take those cardiac systems, combine them with a liver, and in this is what a chip looks like here. We have a recirculating fluid here between these two reservoirs. We did a project with AstraZeneca that was published in Nature Scientific Reports last year. We wanted to look at terfenadine, fexofenadine. Terfenadine is cardiotoxic, fexofenadine is not. We look at a cardiac-only system, we see toxicity from the terfenadine, but when we add in liver, we see it converted to fexofenadine and toxicity go down substantially. Conduction velocity is really not affected in the presence of liver, but in cardiac only, we can see a IC50 of about 33 micromolar. Again, going back to that whole point, we don't see any decrease in viability. What we're really looking at is function, things you'd want to be able to look at during a clinical trial. Also, the systems, what we want to try to be able to use them for is to predict in vivo or clinical outcomes. So to be able to do that, we have to have a PKPD model of the in vitro system, so we can couple that to a PKPD model of the in vivo system. So we build these models, okay? We can actually look at absorption of compounds in the system. We can build that into our model. We can look at mixing, we can look at loss, and be able to get the area on the curve or the exposure of each organ in each system. We can model that and show that this is fexofenadine, which doesn't absorb much in the housings, terfenadine that does. We're looking at the HPLC monitoring of that and see our models can predict quite well. And then when we then couple it into an in vivo model, again, this is with AstraZeneca's data for guinea pig, dog, and non-human primate, you see that we were able to predict quite well the data they had measured from the primates, the non-human primates. We did another project with Roche Pharmaceuticals where they said, okay, can you take your cardiac liver system and not only just look at toxicity, but also efficacy in the same system. So we looked at a non-multi-drug resistant cancer and a multi-drug resistant cancer to be able to look at efficacy and then be able to look at cardiotox and liver tox in the same system. We want to look at tamoxifen. Tamoxifen in the non-multi-drug resistant cancer by itself is not very chemotherapeutic, but its metabolite is, so we add liver in, it shows the decrease in proliferation. The metabolite by itself is about the same amount. And in the multi-drug resistant cancer, what we see is tamoxifen with liver, the metabolite did not work. But then when you put in another compound, verapamil, to block the pump that makes it multi-drug resistant cancer, you see a decrease in proliferation, but no additional decrease in proliferation. So it's not the verapamil acting as a chemotherapeutic, it's actually blocking the channel where we can look at a drug-drug combination in the system. We can also look at the drug-drug combination in terms of the off-target toxicity. Here where you see the control, you see the conduction velocity, you can see it go down, but come back up with tamoxifen only. But with tamoxifen plus verapamil, it goes down but does not come up. Same thing with force, it goes down but comes up, not quite the same way, beat frequency, but again, we're not stopping cardiac viability. So if you're looking at this system, you say, okay, I would probably have to come up with another compound besides verapamil to block that channel, or I'd have to actually let the drug clear for longer before I would do a second dose. And again, the liver was not affected in the system. And again, this was published in Science Translational Medicine last year. We can also look at neuronal systems in our models. This is a two-chamber system where we look at human motor neurons and human skeletal muscle. We try to go the axons through the tunnels. You can clearly see here the axons going through the tunnels, innervate the myotubes, nice neuromuscular junctions. We are able to get a dose response curve for Botox. Currently, in the clinical setting, the IC50 for the dosing is done figured out in animals. We can actually then replace that usage because we have a human model can actually do dose response. We're also, again, interested in rare diseases. In this case, we've been able to differentiate out all the different mutations for ALS motor neurons, FUS, SOD1, TDP43, and C9. And again, they work just as well in the systems. We get nice neuromuscular junctions. And we actually can look at, see there's a difference in the number of neuromuscular junctions, their stability. But again, trying to find something that correlated what you see in a clinical trial, we asked somebody who's running one, okay, what are some of the features that you look for? So well, one of the things we try to do is have someone do a task faster and faster. And what we see is they start to fail as they do it faster and faster. So we can actually reproduce that. We can start stimulating the motor neuron at higher and higher frequencies. So what we see with the FUS and SOD1 neurons, so the SOD motor neurons don't mature as fast, but they still drop off at the higher frequencies. Now, they both get about the same maturity in day 14, but again, it drops off a little bit, statistically significant. But then SOD starts to fail quickly, and then the FUS starts to fail as well. And again, you see it's very dependent upon the frequency increase as to how much skipping you get. We can also look at cognitive function. We're trying to look at long-term potentiation, which is a correlate for learning and memory. We can make circuits out of cortical neurons. These are human cortical neurons, where we have these axons going back and forth between different electrodes and different pools of neurons to be able to look at long-term potentiation. We want to try to be able to look at this in terms of, you know, tox, off-target tox, but also for efficacy. We're looking at really efficacy for Alzheimer's disease. So we then treat these systems with A-beta oligomers, and so that the scrambled oligomers, which means they don't have the same amino acid sequence as the A-beta, we can induce long-term potentiation, and that doesn't go back down when we treat it with the oligomers. But with the A-beta oligomers, we actually see an abolishment of long-term potentiation. And again, same thing with tau aggregates in the control, no loss, but we do see a loss. And so we can actually start looking at drugs that would be affecting things in the mild cognitive impairment or even pre-mild cognitive impairment stages of Alzheimer's disease. We've been able to combine all these together into 4 and 5 organ chips. Again, we have the functional measures of the systems. We publish these in Nature Scientific Reports, Advanced Functional Materials. The latest one in Advanced Functional Materials, we able to show we actually get all 4 organs functioning properly for 28 days. 28 days is a very important time frame for looking at systemic tox. We've also just published the system by adding an immune component where we had cardiac, liver, and skeletal muscle. You see the monocytes flowing backward and forward here across the electrodes, okay? And it's actually, as it pumps back and forth, we see them going. And the system itself does not activate the monocytes, but we then specifically damaged one tissue, and we saw the monocytes only go to that tissue, which was cardiac. But we saw also the emergence of a restorative phenotype, the M2 phenotype, whereas then we stimulated the entire system with bacterial phenotype, looking at trying to simulate something like sepsis. And we saw the complete activation of the monocytes, and attacked all the different organ systems. So again, we have a nice model system for looking at cytokine storm, okay, because we can actually model the innate immune system. And again, this was just accepted for publishing in Advanced Science. Lastly, we have barrier tissues for the system. We can take skin into the system and skin mimics, because we worked with L'Oreal for years. Again, a 5 organ system. We can put in kidney cells, not as a functional kidney, but as a tox target. We can also have a functional blood-brain barrier. Very high tier values, means they're very tight junctions. And actually looking at transport, you can see that the transport in vivo and in vitro is about the same. And also a GI tract-based system, where again, you can actually get very nice tier values. Again, they should be about an order of magnitude lower, which is back to what you've seen. A lot of these systems were developed in Mike Schuller's lab at Cornell, but they've been transferred to Hesperos. So lastly, we have a large number of rare disease capabilities at Hesperos. And this becomes really important because there's 7,700 rare diseases, but only 400 or so active research programs, because they just simply don't have a model. But what we've actually shown, we can actually take induced pluripotent stem cells from patients, derive the tissues that have the greatest effect, build a phenotypic model, and then treat it with various potential therapeutics. And if we can actually get the function back to close to what with the wild type or the normal human, that the FDA will accept this data. So they will accept this data for going into the clinical trial. And so if you want to work with Hesperos for rare diseases, if you want to work with us to try to get your systems more quickly into an IND and cheaper, we'd love to talk to you about it. One of the things that we're really interested in doing is working directly with medicinal chemists, because our systems can work with milligrams of quantity. If you've gotten to the point where you've gotten 4 or 5 different tool compounds that you're looking at, or variations of your single tool compound, you basically have to guess which one is going to actually be your best candidate before you scale it up into grams or kilograms. What we can do, we can actually test 4 or 5 different variations of the tool compound, okay, for therapeutic index, both efficacy and tox for your major toxicity targets in the same system. We're very interested in working with you at that. So if you're interested in talking to us, contact us during the one-on-one Partnering with Bio, or you can also contact us through these different ways before, either coming into our website or calling. And we're quite happy to either talk to you during Bio or to give you a custom webinar after the Bio meeting to get much more in detail into our systems. Thank you very much.